Now, that's a brief introduction to neurons. We'll go into a lot more detail later. I'm going to spend some time quickly going through the glia. I want you to know astrocytes, the microglia, oligodendrocytes, Schwann cells, and radial glia. Okay, so here's a cartoon showing you the relative size of all of these different things. So here we got a neuron. Okay, here we're going to have an oligodendrocyte. Okay, again, I'll talk about what these things mean later. Here we have a microglia. Okay, astrocyte. Now let me give you a couple hints. Cyte means cell. Okay, so anytime you see cyte, you need to think cell. If you can break things down into the roots, things will be a little bit easier. Oligo is going to mean a few. You may have heard of an oligarchy. Dendrocyte, dendro has to do with feet. Okay, so literally an oligodendrocyte is going to be a cell with a few feet. And if you look at the oligodendrocyte, here's the soma and here's the nucleus. There's going to be processes coming off of it, and these are going to have a bunch of different feet. Okay, these feet are actually full of fat. So this is basically the cell that makes the myelin in the central nervous system. Okay, so it's important. Oligodendrocytes are going to make myelin within the central nervous system. Okay, but you can see why they are called a cell with a few feet. Micro means small. Glia, you know what a glia is. So these are small glial cells. Okay, and again, if you compare them to the size of a neuron, you can see they're much smaller. Astro has to do with stars. Cyte has to do with cells. So no, these aren't cells that go out and explore space. But to the early anatomists, these things looked like stars. So they called them literally star cells, astrocytes. All right, I'm going to start with astrocytes. Clinically, astrocytes are going to turn out to be one of the most important cells. We call ourselves neuroscientists. Right? So the field focuses on neurons. It's turning out, and by the time you teach this course, I think you're going to see that actually glial cells, and especially astrocytes, are going to be involved in a lot of psychopathology. It's not just going to be neurons. Astrocytes and other glial cells have profound effects on how neurons function. Here, we're going to use a technique called immunocytochemistry. I'll talk about that later. These cells are going to be stained with a special dye so that we can see something called GFAP. You don't need to remember that, but that's glial fibrillary acidic protein, GFAP. And this is something you only see in astrocytes. Okay, so here you can see in brain tissue what an astrocyte looks like. Now, neurons don't stain because they don't have GFAP. One of the things you can see here, this is a blood vessel. Okay, so here you can actually see the astrocyte cell body sending a process to surround the blood vessel. And I'll talk about that in a minute when I talk about the blood-brain barrier. Astrocytes provide some of the structure that makes up the blood-brain barrier. Okay, and here again we're going to have an astrocyte stained green and a neuron, in this case, stained red. You can see that obviously the two are closely associated with one another. Astrocytes are going to be important for helping to regulate the cell environment around the neurons. Neurons are really picky. They're very easy to kill. They require a very specific chemical environment, or they don't do well. In addition, when we talk about synapses, it's going to be important to remember that astrocytes, here in green, are going to have processes surrounding synapses. And what this allows them to do is they can pick up extra chemical and bring it back in so they can get rid of it. They can help end chemical communication between two cells. The other thing to remember is that they can release their own chemicals, and that can have an influence on how this connection works between two different neurons. And we'll talk about glutamate and stuff like that later. But it'll be important to remember that astrocytes can release glutamate. Microglia are very small. These remove waste materials. They eat viruses, fungi, microorganisms. And if there's damage, say after a stroke, you're going to see an increase in microglia. So here, the green cells, these are going to be microglia. Okay, and this is an animal model of a stroke where they killed an animal various times after a stroke. And you can see before the stroke, not that many. But after the stroke, at two different time points here, post-stroke, you can see many, many more. And that's to help the brain cope with the damage that's occurred. Okay, so microglia you can think of in some ways as kind of the sanitation workers of the brain. I already told you about oligodendrocytes. Okay, these are going to be important for the central nervous system myelin. Okay, so if you lose oligodendrocytes, this myelin goes away. If I kill one oligodendrocyte, how many beads of myelin do I lose? Yeah, I lose a bunch. Okay, you may have heard of multiple sclerosis, MS. MS is an autoimmune disease where the immune system attacks and kills oligodendrocytes. Okay, so we'll talk a little bit more about what that means in a bit. Here's an actual picture of an oligodendrocyte. Okay, here again you can see it stained this light green. These are axons from neurons, so you can see the processes come out. Now we know, not from this picture, but from other work, that this is actually going to make a bead of myelin that covers and insulates that axon. In the peripheral nervous system, the PNS, peripheral nervous system, your axons are also myelinated. Okay, but instead of being made by oligodendrocytes, these are going to be made by Schwann cells. Okay, so one Schwann cell makes one bead. Okay, so one cell equals one bead. So if I kill a Schwann cell, how many myelin beads do I lose? That's right, one. Remember, if I killed one oligodendrocyte, I lost several myelin beads. Okay, this is a cross section, okay, through the cable. Here is the axon. Okay, now here you can see the nucleus of the Schwann cell, and basically what happens is this sends out some membrane, and it's going to wrap around the axon over and over. Okay, so here you can see this kind of wrapping around. This is all from this one Schwann cell. This then fills with fat, and now we've got our insulation. Okay. Now we'll talk a bit about radioglia. These are key in development. Okay, so in adulthood, you don't really have radioglia. In prenatal environment, while you're developing, you've got lots and lots of radioglia. These basically guide neurons. Again, when I talk about development, I'll come back to this. But basically, all of your neurons are made in one place. After they're born, they somehow have to migrate to the right place in the brain. Okay? But right now, I just want you to know that it's radioglial cells that are going to provide the roads that neurons are going to follow. Okay, so you can actually watch, it's amazing, there's videos of this, where you can watch these neurons physically move along these fibers made by the radioglial cells. Right, and again, back to this picture, right? Is this part of the brain or part of the spinal cord? Well, brain, yeah, one is, you can look and see this picture, and you may recognize that as a brain. The other way though, is oligodendrocytes, right? These are only in the brain, not in the periphery. Okay. I mentioned, again, astrocytes, in addition to regulating the synaptic environment, are going to surround blood vessels. Okay? And in the next video, when I get to blood-brain barrier, I'm going to talk about that. Okay, so again, you want to remember astrocytes, oligos, Schwann's, microglia, and radioglia. You'll want to be able to recognize them and tell me briefly what they do. Okay, so that concludes our real quick whirlwind introduction to the cells of the central nervous system and the peripheral nervous system. We're going to talk a lot more about these later. So if you need to, you can always refer back to this quick video. All right, thank you.